All right. Good morning, good afternoon, good evening, everybody. Can you guys hear me? Okay, great, excellent. Okay, thank you for confirming. So welcome to the fourth PEDS Protein Engineering and Design webinar, organized by the journal Protein Engineering Design and Selection. My name is Roberto Chica. I'm the editor-in-chief of the journal. And it's a pleasure to see so many of you here today. So I'd just like to start by giving some information about the journal, and then I'm going to give you some details about how this webinar will run. And finally, I'll introduce the speakers, and we'll go and listen to very interesting talks. Okay, so many of you have already heard me tell you about the journal, so I'll just like to mention that this journal has been around since the 1980s, and it's always been a community-run journal, so founded and run by protein engineers and designers such as yourself. So because of this, I would like to remind you that we're always happy to hear your ideas, your suggestions to make the journal better. And also, we have a lot of new initiatives that if you're interested to know more, I've written in the editorial that's freely accessible on the journal's website. So I'll just mention one that may be of particular interest to you. We have a hot topic special issue that will be coming out in the first quarter of 2021 with many review articles on current topics in protein engineering design and evolution. So there should be something for everyone out there. So these will be coming out in the early 2021, and I encourage you to read them, that they will be made freely accessible to the whole community. Also, I'd like to mention that given the positive feedback that I've been getting from these webinars, I've went ahead and organized the next round of webinars, which will be starting in January. And again, I've been fortunate to be able to convince excellent and top researchers, such as those who are presenting today, to accept the invitation and present. So I'd just like to maybe share my screen with you to show you the lineup, just as a preview. I'll bring it back towards the end in case some people haven't joined yet. So I'll do that right now. Hopefully this will work. Okay, so hopefully you guys can see my screen. There's no way for me to know whether you can, but as you can see from the lineup here, we have already 3 out of the 4 webinars confirmed with speakers indicated here. So we have in February, Dec Wolfson from the University of Bristol. In March, David Baker from the University of Washington. And in April, Elizabeth Meyering from the University of Waterloo. So this will be posted on the PEDS website, of course. But I also would like to remind you, this is the Twitter handle for the journal. This is where we post all the news. So if you want to know about what's going on at the journal, including the webinars, I encourage you to follow us here, because all that information is indicated there. So as you can see from the lineup, it's going to be as exciting as the webinars that we've been having up to now. Okay, so hopefully I'm back. Okay, there we go. Okay, so hopefully everybody was able to see that. I'll just finish by explaining a little bit how the session will work. So we have 2 presentations today. The first one will be a shorter presentation of about 20 minutes, followed by a 5-minute question period. If you have questions, please use the Ask a Question button that's at the bottom middle part of your screen. You can type your question there. The presenter, the speaker, will be reading the question at the end and answering it there. You could also use the chat box, although questions asked in the chat box can be, sometimes they disappear if there's too many of them. So I encourage you to use the Ask a Question button. After that first question period, we'll have our keynote presentation, which will last about 40 minutes, followed by a 5 to 10 minute question period. And typically we are done in a total of 90 minutes. And we've always been able to keep on time. So that's how it's going to work. So hopefully everything will go great. All right, so without further ado, I'll introduce our first speaker, Professor Amelie Stein from the University of Copenhagen. And Professor Stein is from Germany, but she studied in Spain, in Barcelona more specifically. at, I'm going to try to pronounce this correctly, Universidad Pompeu Fabra. And there she worked on the study of protein interactions, so network biology, and using computational approaches to predict and analyze protein interactions. From there, she moved to the lab of Tanya Kortem at the University of California, San Francisco as a postdoctoral fellow, where she did a lot of interesting work developing computational methods for modeling dynamic protein regions, so ways to encompass the different conformations that can be found in these positions. And she also worked with high-throughput yeast screening assays to assess the function and molecular specificity of the SH3 domain family. After this postdoctoral fellowship, she moved to Copenhagen in Denmark as a postdoctoral fellow and now as an assistant professor. And in her group, she studies the effect of mutations or disease-causing mutations. And this is some of the work that she'll be telling us about today. So it's a pleasure to have you. Dr. Stein, please take over, and we look forward to your presentation.
Yeah, thanks so much. Let me start by sharing the screen. Thanks so much for having me. This is really exciting. And as Roberto already pointed out, so in the last few years, I've become really interested in the question of variants that we observe from genomics and what their consequences to proteins and ultimately our bodies might be. And so that's also what I'm going to talk about today. I hope you can see my screen. Essentially, the motivation, as I already was saying, comes from this data avalanche that we have from genome sequencing. So we now know that each of us carries thousands of variants in our DNA, but which of those have a clinically relevant consequence, such as increasing our risk for particular diseases, and why that happens on a cellular and mechanistic level, that has remained an open question. And so in all of this work I'm talking about, we're focusing on missense variants in proteins, so single amino acid changes, in part because coming from protein science, this seemed to be the area where I felt like, oh, this is where I can make the most contribution. We already have frameworks from protein design, for example, for thinking about how do variants affect our protein structure and function, but then also looking further ahead, if we understand what goes wrong with any particular protein in its environment, then we could use this as a starting point for therapeutic developments, and I'll get back to that a little bit towards the end. But so to start out from, I would like to introduce the sources of data that we're working with. There's two large databases we're building on. One of them is ClinVar. It contains data from patient samples. And so in the ideal case, this data has also been classified by experts. So for example, people working in clinical genetics that know, oh, this particular variant is seen in a lot of patients that develop, say, a particular type of colon cancer, and it's never really seen in people who do not develop this disease. So this is considered pathogenic. And then on the other end of the spectrum, we would have benign variants, so those that are observed occasionally, but that have not been associated to cause any disease. So these are the two ends of the spectrum. Actually, the vast majority of the data is somewhere in the middle. So they have been seen in patients, perhaps also patients with disease, but it's not clear whether there's any causal relationship to those diseases, or if it's just an observation by chance. And so on the other side, we have the NOMAD database that stores aggregated data from population sequencing. At this point, there's data from over 100,000 individuals. And we're using this mainly as a, you could call it background knowledge. The reasoning being that anything that's common in such a large cohort is unlikely to cause severe disease. And when you look at NOMAD, one striking observation to me is that the majority of variants are singletons. So they've been observed exactly once despite sequencing so many people. And I think that's already a good illustration of why we need general models and can't just have a lookup table where, oh, this particular variant in this particular protein causes a disease or not, simply because we won't observe them often enough to fill out such a table. And now when we started out with this project and wanted to know, well, how do variants lead to pathogenic consequences? We picked a protein that has quite a number of pathogenic variants. it also has a known structure and function. So here we have MLH1. It's involved in mismatch repair, a kind of DNA repair. And pathogenic variants in this protein are associated with early onset colon cancer. So where individuals carrying these variants, they develop colon cancer in their 30s or 40s. And we know it has a well-formed N and C terminal domains. There is a linker of about 100 residues that we don't know that much about. It's likely disordered. It does not contain any of the pathogenic variants. And so we looked experimentally at about 70 variants scattered throughout the two domains. And what we observed is that quite a number of them have lower levels than the wild type. And I'm not going into any details of this here. You can read it up in the paper. But essentially, we could show that these lower levels are because those variants are degraded. So the cell has a quality control system that detects unfolded proteins, whether fully or partially unfolded, and tags them with ubiquitin for degradation. And so this process leads to the lower levels of many of these variants. And now, in computational modeling of proteins, we have tools, for example, Foldex or Rosetta, that are able to calculate this folding energy. And if we apply this to both the wild type and the variants we're interested in, then that can give us an estimate of whether we think these should be, based on the protein modeling we have, these should be destabilized. And indeed, we observe between these delta-delta-G calculations and the cellular protein levels, we observe a reasonable correlation. It's not perfect, but so we see for the benign variants that have the protein levels around the wild type, around 100%, we also see that their delta-delta-G is around 0, so we predict them to be neutral with respect to stability, and that's also what we see experimentally. And then there's quite a number of pathogenic variants that have lower levels experimentally and that have a high delta-delta-G, indicating that we expect them to be destabilized. Now, one advantage of this computational modeling is that we can much more easily scale it up, which is not really the case for many of the experiments we're doing. And so here we're comparing the change in protein stability, the delta-delta-G, for all variants of MLH1 that have been reported in NOMAD. We plotted here on a log scale. There's very few that are really common, and most are quite rare. And so again, we see that the benign variants, they're all effectively neutral with respect to stability, while there is a substantial group of pathogenic variants here which have high DDGs, and we indeed expect them to be destabilized and also to be pathogenic because of this degradation process that I explained earlier. But at the same time, we also see, oh, there's quite a number of variants that are listed as pathogenic, but they appear to be neutral with respect to stability. And so we certainly know that stability isn't everything it takes for a protein to be functional. And in this case, we're lucky to have not only this cellular protein levels, so experimental data of how much of each of the variants there is in the cell, but also data on in vitro activity from this earlier paper from a Japanese group. And we also have data from cellular assays of whether these variants are still capable of the mismatch repair that they typically should carry out. And so we do see that there's about two groups. One where activity is essentially wild-type-like, cellular protein levels are essentially wild-type-like. They're also functional, most of them, in the cellular assays. And we have another large cluster where levels are low, activity is low, and they're non-functional. So that's all quite consistent. And then we have this sort of side cluster here where the levels are high, so stability does not seem to be affected, but nevertheless, the in vitro activity is low, and some of those also were not functional in the cellular assays. And those are variants that are close to the active site of the enzyme. So these are changes that do not affect the protein stability, but nevertheless are detrimental to its overall function. And then plotting the ClinVar designations here back onto this, we see, as we would expect, that the benign variants are up in this quadrant, where we have high levels and high activity, while most of the pathogenic variants are here, where levels are low and activity is also low. And to have a handle of also capturing computationally these changes to the protein function that may or may not have anything to do with its stability, we turn to analysis of sequence conservation and its context. You may have come across this in protein structure prediction, where it's been used quite successfully to identify contacts, but it's also been used by us and others to recognize pathogenic variants. And in analogy to the delta-delta G calculations that we do for stability, we're referring to this as delta-delta E. And so when now we take the delta-delta G, the calculations we've had before, and combine them with the delta-delta E, we see that most that are high in delta-delta G, so that are destabilized, they're also high in delta-delta E. And one thing this indicates is simply that evolution favors stable proteins. So all those here, effectively, they have been selected against. And we could say that we have one category of variants here, where there likely is loss of stability, and they will be degraded, and this degradation leads to lower levels and effectively to a pathogenic or loss of function phenotype. And we have this other group of variants where many are detrimental. Those are stable. They appear neutral with respect to stability, but other functions are affected, like, for example, the ligand binding or also binding to other proteins. And so overall, we were quite happy that this worked well in classifying variants of MLH1. And we're wondering, is this a generally useful approach? Can we use these calculations to classify especially pathogenic variants or also later unknown variants from other proteins? And so we made a medium scale data set of 18 proteins. They were involved in different diseases, so some associated with hereditary cancer risk, others with neurodegenerative diseases, and lastly, there's a group of metabolic syndromes that are affected. And for all of these, we carried out the delta-delta G and delta-delta E calculations, and we see that, quite similar to what we observed already for MLH1, that there is a quite different distribution of where the pathogenic variants are in this space spanned by the delta-delta G and delta-delta E versus where the nomad variants from these proteins are. So those found in the population, they're typically in this region here, where both delta-delta G and delta-delta E indicate that these changes are well tolerated, while the majority of pathogenic variants are here almost on the other end of the scale. Because this visualization doesn't lend itself very easily to capturing what's the largest group, I've transformed it to this other image where you see that indeed the group with the high delta-delta E and delta-delta G is so destabilized and also rare across the protein family. That's the largest group of all those pathogenic variants. And this is for the whole data set of 18 proteins. But now if we split it back into the individual proteins, we see that there certainly is variability as for how important the two different factors are. So for example, here for MSH6, like loss of stability could explain up to 95% of the pathogenic variants. So that seems to be really a crucial factor. While on the other end of the distribution in this protein, ADSL, there's only around 60% that are of the pathogenic variants that are destabilized. And then you also see that for a number of the proteins, there are variants that are classified as pathogenic and where neither of those two approaches is able to pinpoint even that this would be problematic. I'll get back to those a little bit later. And as I told you earlier when I introduced ClinVar, the majority of data that we have are actually variants where the significance, the clinical significance is not known. So they've been observed in patients, meaning there is more of the background risk. So it's different from nomad in that these variants are in ClinVar because many of them have been seen in patients, but it wasn't clear whether this variant is causal or whether it's a coincidence that a person develops a particular disease and also carries this variant. And so since now we have two scores, two predictors that can give us some information on what happens in these proteins, we've applied them to all the variants of unknown consequence in these 18 proteins we've been looking at. And the first insight from this, I think, is quite reassuring because it shows that around 50% of the variants of unknown consequence appear to be neutral. So they're certainly neutral with respect to stability, and also the sequence alignment indicates that these are likely well tolerated. We can't exclude that there are very specific mechanisms, such as changes to phosphorylation or splicing, that would render them pathogenic, but overall, it's much less likely than for these other variants in the other groups. And so then we have this group over here, where the DDG is high, the delta G is high, so these variants have a high chance of being degraded, leading to lower protein levels. And for example, if one wanted to do a follow-up study into specific variants here, I would definitely recommend looking at degradation patterns, and potentially, when looking for treatment, also for rescue potential of these degraded proteins. And then there's this other group here of about perhaps a quarter of the detrimental variants, and those appear neutral as far as stability is concerned. So those are much more likely to affect binding of ligands or other proteins. So to summarize, I've shown you that many of the pathogenic protein variants have high delta Gs, so they show loss of stability, and this makes it very likely that degradation and resulting low levels of proteins in the cell are the mechanism that makes these proteins pathogenic. And moving forward, it's an interesting opportunity to think about stabilization or prevention of degradation of these variants to alleviate those diseases. And the most famous case where this is already successfully being done is cystic fibrosis, where at this point, 90% of carriers of pathogenic variants can be treated by existing drugs. And we've combined the sequence analysis, the delta E, with the stability changes, and we find that together these are helpful in pointing out the likely mechanism that underlies pathogenicity for individual variants. And we've been quite happy with how well this works, but of course, there's always room for improvement. And so I'll give you two quick illustrations of that. So one is, again, this plot of the population variants in the background, and then as dots, I've indicated those variants that are classified as benign. And many of them are here in this area, where both metrics say these are likely neutral. That's what we would expect, but there's also quite a number that get fairly high scores, and that's definitely an opportunity to look into whether our modeling is going awry. For example, some that get the highest scores in delta G are mutations that involve glycine, and we know that we're not as good at predicting those as well as some others. And in general, if you think about how the tools, so Foldex and Rosetta, for example, were parameterized, much of this data came from Protherm, which is a resource that has collected years and years worth of biophysical characterization of protein variants. And this has been tremendously useful. But if you look at the distribution, you can also see, oh, a lot of this came from alanine scanning, and there's a number of possible changes that we simply don't have data for. So it's quite possible that we're doing more poorly on those in the light yellow or white areas because we don't have data to even train our parameters well. And now it's possible to do multiplexed assays in a way that essentially provide as much information as we have in Protherm, but in a single experiment. And both ours and also other groups are working on generating such data, some of it specific to stability and other that also involves function, to hopefully improve the modeling we can do. And with that, I would like to thank the people that are involved in this work. I started this when I was a postdoc in Heston's group, and we're still collaborating on this also with Rasmus Hartmann-Peterson's group, and the people working with him, especially Amanda and also Sophia, did much of the experimental work I showed you. And we have this PRISM Center where also Doug Fowler, Michele Minuscolo, and Tom Hansen are involved, where we're generally very interested in the different angles we can take to understand what's going wrong with protein variants. And thank you very much for your attention.
All right, thank you, Amelie. That was very interesting. Okay, there's already a question that's in the Ask a Question button at the bottom.
For the stability calculation. Okay, I'll just read out the question. For the stability calculations, the mutations that you're probing for the benign and pathogenic variants are located anywhere in the protein surface core. How do you deal with the relatively high accuracy of the prediction tools for surface exposed or residues? That's a very interesting comment. It's definitely an important factor. My observation is actually that accuracy is better, at least in this sort of semi-quantitative way of looking at protein variants. We're okay with knowing, oh, this is destabilizing, and then whether the score is a 5 or a 15 or a 250 doesn't really matter so much. We actually find that our predictions work better in the core, and that we're struggling with the surface variants. But I have also seen in other papers, for example, Steve Mayo's, that the observation is sometimes the reverse. So I think there's clearly work to be done. I've done this same analysis, and it's going to be in the preprint that's hopefully out soon, split into core and surface. And yeah, we find that we do better in the core, but it's definitely an open issue. Next question. Can you comment on mutations of a given protein-associated disease that may fall in different quadrants in the plot? And what's the mechanism? Yes, can you still see my slides? I sort of assume you can't.
No, we can't.
Oh, okay. I'll try to give you the visual impression then. So if it's in the quadrant where the delta delta G and the delta delta E are high, then we think this is where we're the most confident that this is detrimental, because both evolution and what we know about protein biophysics tell us that this is going to be a terrible idea. And so there, we do think that these are going to be destabilized. And in that sense, they're not, we couldn't mechanistically, it's very hard to say the difference between those and those where evolution says, oh, this is going to be fine. There's very few where Foldex or Rosetta will say, oh, this is terrible, and evolution will say this is fine. This is very rare, I think, because evolution favors stable proteins, but I don't have really hard data to prove that. And then there is the other two. In any case, essentially, those with a high DDG, we would say there's a high chance that these are going to be degraded, and that's the mechanism that renders them pathogenic. And then there's the other side, so where the DDG is low, and if then the delta delta E tells us this is a bad idea, evolutionary speaking, then often we've also mapped these back to the structure. We find them close to, say, the active site or protein interaction interfaces. And then there's the last quadrant where both say the score is low, and that's where we usually find variants that are neutral. So that's where we find most of the benign variants, and we find the vast majority of anything found in population sequencing. We occasionally find other variants that are still pathogenic, and that then tends to be, for example, splicing sites. So where actually the change in DNA leads to a change in splicing. So the protein we're doing modeling on is not the protein that the cell produces. And so that, I think, is also an important insight, meaning that we need to integrate the DNA level and the protein level. But that in itself isn't actually an issue with the protein modeling. It's more the translation we do from the DNA level to the protein level.
If I may, I would like to ask a question. So
Please.
your delta delta G calculations imply the use of a protein structural model. Am I correct?
Yes. Yes, you are.
Some of these proteins may undergo conformational changes, and you may have access to more than one crystal structure for that particular protein in the PDB. And the effect of mutations could be predicted to be different depending on which state they're modeled in. So is that something that you've considered and that you would want to incorporate into your approach?
It's certainly a very good point. We have considered it only theoretically, so in the sense that we're aware of it. I have to say, for most of these proteins, there is only a single structure, so in practice, it hasn't come up. We have worked on a protein called PEDS that's involved in a metabolic disease, and there we've tried with different structures that are in different conformations, and honestly, it's been a bit all over the map, so I would say there perhaps the errors that we have in the calculations still outweigh the benefits that we got, at least in that particular case, from using multiple structures. But in principle, sort of moving forward, that's definitely true. And I guess the other side of it is, oh, are we limited to applying this to proteins that have a structure? And fortunately, it's not quite as bad. So we have, and that's a separate work, but we have tested a number of different protocols and found that there is one Rosetta protocol that's called Cartesian DDG, and that does quite well on homology models. While the classic DDG monomer and also Foldex are quite sensitive, and they work all equally well on crystal structures, but then if you move into homology models, I would strongly recommend using Cartesian DDG.
Right, and there's also the Flex DDG protocol.
Yes, that we haven't benchmarked because I think that came out after we did most of that at the study. But yes, that's also an interesting point. That's true.
Okay, great. I'll also mention that Steve Mayo has made a comment here. He says the Profer database no longer available. All of the data has been transferred to the Protobank database. That's true, yes. And anybody can upload their data to Protobank. So if you have mutational data, you can put it in there, and it's a valuable resource for the community. So I encourage you to check it out if you haven't played with Protobank.
Though, you know, I saw a paper that came out, I think in NAR, maybe last week, that was Protherm revisited after 15 years. Maybe I can post it in the comments. So maybe Protherm is up again. I have not actually checked the URL, and it's true that there's a lot of valuable data in Protobank.
Right. Okay, great. And you have a final question in the comments there.
This may sound obvious, but have you made any observation regarding the length of the polypeptide chain or the oligomeric state on the chances of accumulating destabilizing mutations? I would say not specifically. So we've definitely found, we have not, maybe we also haven't looked at it systematically enough, but we definitely see small proteins where there's a number of destabilizing variants. And we also see this for large proteins that we have a couple of homotetramers where we see the same effect. So if there's anything, it is not super apparent, and we have not looked at enough data to make that conclusion.
Okay, great. So thank you again, Amelie, for the nice presentation.
Thanks so much.
The nice discussion. Okay, so now we're going to move to our second presentation, our keynote presentation, that will be given by Professor Donald Hilvert from the ETH Zurich. So it's my pleasure to introduce Dr. Hilvert. He obtained his PhD in organic chemistry at Columbia University, working with Ronald Breslow, after which he undertook a NIH-funded postdoctoral fellowship with Emil Kaiser at Rockefeller University. And then he started his independent career at the Scripps Research Institute, where he rapidly went up the ranks and became a full professor there, but shortly thereafter moved to the other side of the ocean, to ETH Zurich, where he's been since 1997. And Dr. Hilvert, many of you know him. He's contributed a lot of very important work in the field of protein engineering and design. I'll mention just the work in enzyme engineering and design, which I think is really interesting. So he was involved in all of those groundbreaking de novo enzyme design papers from many years ago, and contributed a lot of interesting analysis of what made them good or poor catalysts, depending on the variants, and studying this by directed evolution. And what I really appreciate about Dr. Hilvert's work is that he's very interested in answering this fundamental question of why are enzymes such good catalysts? And I think this is a really crucial question in protein science. And I think Dr. Hilvert has contributed a lot of really valuable and important work in this field. And I know myself, I benefit from that information in my own research. So it's a pleasure to have you here. And today, Dr. Hilvert will be telling us about his work in enzyme engineering and design.
Thank you, Roberto. Can you see my screen?
Yes.
Okay. So it's a pleasure to be with you today and to be able to tell you a little bit about our recent work on metalloenzymes. As probably most of you know, biocatalysis today is an increasingly attractive option for making pharmaceutical agents, agrochemicals, biofuels, and the like. Nevertheless, natural enzymes aren't always optimal, and they may not even exist for the certain types of reactions that you're particularly interested in. And for that reason, protein engineers have become quite excited about the possibility of designing new enzymes from scratch in the lab. But that turns out to be a hard problem. However, over the last 15 years or so, by combining computational methods with experimental evolution, it's been possible to create a handful of enzymes that really match the rates and specificities of their natural counterparts. And Roberto alluded to some of the examples from my lab where we've collaborated with a number of computational groups, Steve Mayo at Caltech, Ken Houck at UCLA, and David Baker at the University of Washington on various different projects. So in the first example, the Kemp eliminase, we worked with Steve to create a catalyst that can do a very simple proton transfer reaction. And starting from his design, which Roberto, by the way, was involved in when he was a postdoc at Caltech, we were able to create a catalyst that can accelerate this elementary reaction almost a billion-fold over the background reaction. Working together with David Baker, we've made very efficient, very selective Diels-Alderase and also a highly active aldolase. In the latter case, we exceed a billion-fold in rate acceleration. And what's notable in this study was that during evolution, we generated a series of catalytic residues, a tetrad that is tightly hydrogen bonded and is essential for efficient amine catalysis of this reaction. Nevertheless, these three different systems are all model reactions. The chemistry is fairly straightforward. It's fairly well understood. Where do we want to go from here? And ideally, we would like to move away from model systems to more difficult chemical transformations. One way of accessing that kind of chemistry would be to exploit cofactors or metal-ligands. And the latter is what I'm going to tell you about today, because metalloproteins are ubiquitous in nature. Probably 40% or so of all natural enzymes use metal-ligands. They can be very simple, like magnesium. Polymerases, for example, use magnesium to activate DTPs or NTPs and oligomerize them. Hydrogenases use iron and nickel to do the reversible oxidation of molecular hydrogen. Nitrogenases uses molybdenum and iron for nitrogen reduction and fixation. Cytochrome P450s use iron atoms to rip hydrogens out of unactivated CH bonds. And zinc is very versatile for metalloproteins. So we wondered whether we could exploit this chemistry to do new things. But rather than start with a natural metalloprotein, we had the ambition to create something de novo. And here we collaborated with Brian Kuhlmann. The idea is very simple, and it goes back to a strategy that Margaret Dayhoff published more than 50 years ago in a seminal 1966 science paper. She had argued that the origin of proteins was in small peptides, and metal-ligands could have been very important in this regard because the metal-ligands could essentially affect assembly into supramolecular structures. And then by linking the N and C termini of the two halves, one could fuse these at the genetic level and then subsequently diversify, removing a metal-liand, adding a new activity, catalytic activity, and whatnot. And so we wondered whether we would be able to mimic this process in the laboratory. Brian had already done a great job at the outset. He used the Rosetta Match program to assemble a supramolecular complex of peptide and metal-ligands. He took a natural 48 amino acid polypeptide, which is shown here. he remodeled the entire top face, including introducing 2 histidines per helix. And the idea was that when you mix this peptide with zinc ions, it would assemble into a dimer where the metal ion served as essentially glue between the 2 halves. And the design he called MID1, which stands for Metal Interface Design. It was the first one that they looked at. And experimentally, this worked very well. The peptide was very well behaved, and when metal ion was added, it formed a very stable dimer. In fact, he was able to crystallize it, and the dimer looked very much like the original design, with one important exception. So in the design, if I go back for a moment, he had anticipated that each zinc would be held by 4 different histidines, 2 from each chain. But in the crystal structure, what you can see if we just focus on this metal binding site, there are only 3 histidines. And one of the histidines that was supposed to coordinate is actually pointing away from the metal site and makes a stack with the symmetry-related histidine from the other polypeptide chain. As a consequence, there's an open coordination site, and in the crystal structure, that site is occupied by a tartrate molecule from buffer. But you could imagine that this could become a primitive active site. And so this is where we joined Brian. He had already shown that the assembly step into the supramolecular complex was possible. And Sabine Studer in my lab then dimerized the protein, linking the N and C termini of the 2 polypeptides with a short peptide linker. And then they together diversified this computationally to remove one of the metal binding sites. And specifically, we removed this one on the right, which is the more solvent exposed. Initially, we thought it would be much more easy to optimize a protein if we only had one metal site to worry about, and that's going to be the one that's deeply buried. So Brian had also shown before we started that he could replace tartrate, for example, with an activated ester, paranitrophenolacetate, and see that it could be efficiently hydrolyzed. The zinc can bind a water molecule, lower its pK, generate high concentrations locally of hydroxide that can then attack the bound ester substrate. And so we wondered whether we could take this simple starting material and evolve it into a lipase-like catalyst that could do enantiospecific hydrolysis of these coumarin esters. So we have a chiral center here, and we wondered whether, first, could we make a very efficient catalyst? And then secondly, would it be able to control the attack on the 2 enantiomeric forms of the substrate? Now, the starting catalyst is able to cleave this substrate. It's not a very good catalyst. Here we're looking at a Michaelis-Menten plot of initial rate versus substrate concentration. We can get the steady state parameters. You can see Kcat is 0.01 per second. Kcat over Km is about 20 per molar per second. So it's not very, very active. I mean, in fact, you're just seeing a little bit of hyperbolicity in the trace. Turns out that the catalyst is also enantiospecific. It preferentially cleaves the R-configured ester substrate, but the preference for it over the S enantiomer is only 2 to 1. However, we can use directed evolution to optimize it. And on this slide, you see the normal workflow for my lab. We basically start with a gene and we introduce mutations randomly into that molecule. Here we use various techniques. We can do saturation mutagenesis of residues that line the binding pocket. We can introduce mutations randomly using error-prone PCR. We can combine favorable mutations using DNA shuffling. Making diverse populations of genes is really easy nowadays. This is basically a solved problem, and the method that you choose depends on the question that you're asking. Once you have a population of genes, you use it to transform E. coli cells. Each cell gets one gene. Each cell produces one protein. So if we grow them in arrayed fashion in microtiter plates, we can easily sort out genotype-phenotype issues and specifically identify those variants that have mutations that are a little bit better than the starting clone. So we typically grow the cells to dense cultures. We spin them down, break open the cells, and add our substrate, in this case, look for fluorescence, which is very, very sensitive. So typically we would look in these experiments at 100 to 1,000 clones every couple weeks. So not incredibly high throughput, but certainly sufficient to optimize these low starting activities quite substantially. So Sabine did this. She carried out 10 rounds of mutagenesis and screening, and in the process introduced 20 mutations. The whole protein is only 95 amino acids long, so we've mutated almost 20% of that molecule. And you can see the site of the mutations by these magenta balls, and basically they're distributed in both, almost equally in the two halves of the molecule. This leads to a significant increase in catalytic efficiency. So protein chemists like to think of sequence space as a vast mountain landscape, and if you live in Switzerland, that's a very attractive image. So we can plot transition state affinity, log 1/kts, which is a measure of chemical proficiency versus rate acceleration on the x-axis. And so our starting protein is not very active. The activity is somewhere in the foothills. It's very similar, in fact, to a natural zinc protein called carbonic anhydrase, which will also catalyze the hydrolysis of paranitrophenolacetate, for example. But using mutagenesis and screening, we can increase the activity very, very substantially over these many rounds of mutagenesis and screening. In this case, increasing the chemical proficiency almost 50,000-fold. Turns out that this particular metalloprotein was much easier to evolve than a computational design that we worked on with David Baker that was also supposed to be a good esterase, in this case using not a metal-ligand but a cysteine as a catalytic nucleophile. We just were never able to get this much better than the activity that you see here. This is already the evolved variant. And I think this tells us first that the de novo scaffold is really quite an effective starting point. And secondly, that metal ions provide this intrinsic activity that is very, very attractive. And if we can bring the two together, then we can get really quite interesting activities out. So our best variant has a kcat now of 2 per second, which is quite respectable, and a kcat over km of almost a million per molar per second. This is the apparent bimolecular rate constant for collision of enzyme and substrate. And it's only 2 orders of magnitude down from the diffusion limit. So I think this tells you that this strategy is a very effective one. Note too that the enzyme has become quite enantiospecific. Has a 1,000-to-1 preference for the S-configured enantiomer. So it's opposite of the starting design. And this change in specificity came about even though we only screened with racemic substrate. We never put any specific pressure for one or the other isomers. As the catalyst became more active, it became more specific. And I think this is a very general observation that we've made in other projects in which we've evolved primitive activities. Selectivity is usually comes along basically for the ride. To understand what happened in molecular terms, we needed to obtain structural data, and we were lucky to be able to crystallize our protein in the presence of a transition state analog, namely this phosphonate derivative that you see here in the middle of the slide. The structure is quite good. It's about 1.34 angstrom resolution. And here's a view of the protein with the molecular surface on it. You can see that we have a very deep pocket. The phenylpropionate binds most deeply, and you can just see the edge of the zinc here, bound to the anionic phosphonate group that mimics the tetrahedral anionic intermediate and transition state in the cleavage of the ester. What was striking is that in the evolution, we have observed a dramatic remodeling of the polypeptide chain. So this is what we expected to see based on the original design that Brian had made. with 3 histidines bound to the zinc and 1 turned away. But in the course of optimization, we see this very, very dramatic change in conformation that leads to narrowing of the crossover angle between the 2 helix-turn helix elements. And we believe that this is really crucial for activity because it creates a much larger enclosed binding pocket that can recognize the ester substrate. And see, here you can have a cutaway view of the binding pocket, and on the left you can see this deeply buried ACO component, and it can only fit the S-configured substrate. The R-configured substrate would have strong steric clashes with the back of the molecule. If we turn everything off and just look at the bound transition state analog, you can see that it interacts nicely with the zinc. The zinc in turn is coordinated to the 3 histidines. One of the histidines is backed up by an asparagine that emerged in the course of evolution. This is the kind of interaction that you expect in naturally occurring zinc proteins, and it's used to tune the reactivity of the metal ion. In our case, we also generated an interaction with the phosphonate ligand that was not present in the original molecule. This arginine came along during evolution. You can see it makes a bidentate interaction with the oxyanion and is presumably important for stabilizing the intermediate and the flanking transition states. Note that the guanidinium group also stacks directly on the coumarin leaving group. So we have this cation pi interaction that may facilitate departure of that coumarin alcohol. So if I summarize this part of the talk, what you can see is that it's been possible to recapitulate the presumed biogenetic pathway of metalloproteins in the laboratory. This was easy and it was relatively fast. In the process of evolution, we also converted a structurally essential metal ion. The metal ion was absolutely essential in the original supramolecular complex that Brian had designed. But in the final enzyme, it no longer plays a structural role. It has no effect on the stability of the enzyme. And in fact, it binds relatively weakly in the micromolar range rather than the nanomolar range. But it's become a dedicated cofactor, a catalytic cofactor. Without zinc, we see no hydrolysis. So this is an interesting twist on the normal kinds of effects that you would expect in these systems. It's also notable that during evolution, the structural and mechanistic features of natural zinc enzymes, the activation of the ligands that hold the metal ion, the introduction of oxyanion stabilizing groups, shape complementarity, et cetera, all emerged spontaneously. And as a result, we're able to achieve very high rates and very high stereospecificity. So looking forward, what can we do with this system? Well, in principle, you can imagine continue the evolution. Can we make a diffusion-limited catalyst? We're only 2 orders of magnitude down. But using the screening methods that we used in simple plate assays, it's going to take a long time because you can only really look at 1 or 2 mutations per round of evolution. So we're now turning to droplet-based microfluidics, which we've used very successfully in the evolution of the aldolase that I mentioned at the beginning of the talk. In this case, we can look at 100 million variants in a single day. So you can look at 50,000 variants every second, essentially, or 1,000 variants every second using this technology. So we're very excited to be able to see how far we can push these systems. We'd also like to explore other reactions. We chose ester hydrolysis because Brian had shown that the original MID1 design was able to bind and activate paranitrophenolacetate. But there's no reason that we couldn't imagine binding other kinds of substrates. And we've been particularly interested in Diels-Alder reactions, which are catalyzed in the laboratory using Lewis acid agents. And until very recently, there were no known examples of Diels-Alderases in biological systems. But in the last couple of years, a whole series of enzymes have emerged that are able to catalyze 4+2 cyclizations that give rise to a range of natural products that you see on this slide. These include regular Diels-Alder reactions in which you're making 2 carbon-carbon bonds, as well as hetero Diels-Alder reactions in which either the diene or the dienophile contains an oxygen atom. Most of these natural products arise in polyketide or appear in polyketide biosynthesis. They tend not to be very active as standalone catalysts. And the structural data that's available suggests that that is largely a consequence of the fact that they use very simple mechanisms, a few hydrogen bonding interactions, shape complementarity to speed these reactions up. And so we wondered whether it would be possible to use the MID1 scaffold and the zinc ion that's inside as a Lewis catalyst. And the specific reaction that we targeted is the one that is shown here, in which we have an azo-chalcone, which could in principle chelate to a metal ion. We're going to use zinc, and vinyl indole. These two substrates contain structural elements that are very familiar in many natural products, and so that was attractive as a starting point. In addition, they have the interesting feature that both of them can react either as diene or dienophile. So here you can see the diene, here would be a diene, this would be a dienophile, this could be the dienophile. And they give rise then to a normal Diels-Alder reaction or the hetero Diels-Alder product. And in fact, if you carry this reaction out in DMSO, you get a 1:3 ratio of the normal to hetero Diels-Alder products. So what happens if you try and catalyze this with MID1? Well, you add the two substrates to the protein, and what you find is you don't get anything. There's no reaction whatsoever. And so we turned to our friend Ken Hauk and his postdoc at the time, Gonzalo Jimenez. He's now an independent scientist in Spain. Tackled this problem using Rosetta design. And Gonzalo identified two mutations, a tryptophan and a leucine, that bracket the active site and seem to be able to stabilize the transition state in this binding pocket and block solvent. If we put space-filling representation on the two bound substrates in the transition state in the binding pockets, you can see that this pocket will bring the two molecules together and activate them and increase the probability that they'll collide in a productive fashion. In the event, introducing these two mutations into the scaffold gave us some activity. So in gray, you see the zinc background reaction, and in orange, the new catalyst. Again, it's not a lot of activity. This is a very high catalyst loading, by the way, but it gives us enough of a signal that we can optimize by directed evolution. Here are the kinetic parameters. So we're looking again at initial rate versus, in this case, dienophile concentration. Again, just a hint of hyperbolic behavior, but the kcat is 0.02 per second. The Michaelis constants for the diene and the dienophile are in the millimolar range. This catalyst also has some diastereoselectivity. There's it only produces the dihydropyran. There's no evidence of any of the normal Diels-Alder product. And the stereoselectivity is for production of the 4S6S endo enantiomer, but you can see it's only preferred a little bit over the 4R6R variant. So Sophie Basler in my lab, when she started as a graduate, as a master's student, working together with Sabine Studer, evolved this enzyme over 7 rounds of mutagenesis and screening. In this case, they introduced 10 mutations into the protein, again distributed relatively equally over the 2 halves of the molecule. This led to a dramatic improvement in catalytic activity. So these are the original traces now at lower enzyme concentration. You can see that the catalyst is no different than the background control. But after introduction of those 10 mutations, we completely convert the substrates to product within 10 or 15 minutes of reaction. And we get out only a single product. And it turns out to be the 4R, 6R endo isomer, which was crystallized from the product mixture. This is the control mixture, just again for reference. So we've been able to control reaction pathway. We only get the dihydropyrans, and we can completely control the stereospecificity of the reaction. It's also a very efficient catalyst now. Here the kcat is 10 per second, and the Michaelis constants for the diene and the dienophile are between 100 and 200 micromolar. Now we can compare this again using a fitness landscape, and there are very few of natural Diels-Alderases that catalyze bimolecular reactions, but there are lots of artificial Diels-Alderases that have been designed, including catalytic antibodies, which are shown in black, and ribozyme, which is shown in cyan, and the Diels-Alderase that I mentioned at the beginning of the talk that we worked on with David Baker. This is the original computational design, and after computational and experimental evolution, we were able to push its activity to a reasonably high level. For reference too, there are a number of kinetically characterized natural Diels-Alderases that do unimolecular reactions. And so we can calculate the chemical proficiency, but not the rate acceleration, or in this case, effective molarity, because the molecularity of the reactions is different. But with the Metallo-Diels-Alderase, again, we're starting at very low level of activity, but with mutation and screening, we can improve this really quite dramatically. The best variant, DA7, is 1 to 2 orders of magnitude more proficient than any other catalyst, artificial or natural, that has been described to date for a Diels-Alder reaction. Here too, we have a crystal structure. This was obtained at 1.5 angstrom. Here is the zinc ion. You can see a relatively deep, in this case, hydrophobic pocket. If we remove the surface, we can see the two computationally recommended residues, the tryptophan and the leucine, that bracket the active site. These have been preserved throughout the entire course of evolution, and the other mutations are spread out through the entire structure. Only one actually is in the binding pocket. You can't actually see it because it was a leucine to glycine mutation right at the beginning of the experiments that opened up a big pocket. Another salient difference is the coordination geometry of the zinc. Just as in the esterase, we have completely switched the crossover angle, and that's because the histidine that was originally switched off has been mutated to a cysteine. It's a very good ligand for zinc, and the original histidine that had been bound to the zinc has been converted to a leucine. This locks the protein in the active form, which gives us this voluminous binding pocket for the diene and the dienophile. We don't have a crystal structure with a ligand bound, but Gonzalo was able to dock the transition state into the binding pocket, and you can see that we get a very tight complex. This is the diene. This is the dienophile. The diene is in the back. The heterodiene is in the back, coordinated to the zinc. And where the reaction takes place is completely controlled, completely engulfed in the protein. So if we look directly at the interactions, you can see that the heterodiene is chelated to the zinc, provides a very, very nice interaction. The attacking vinyl indole substrate is held in place in this hydrophobic pocket that was created by removal of that big leucine. And if we look around at the other side of the protein, we can see a very nice hydrogen bonding network that's used to activate the dienophile through hydrogen bonding interactions to the NH of the indole. So we have this glutamine, tyrosine network of interactions. These are the kinds of interactions that you expect in highly evolved natural enzymes, but are still quite hard to design computationally. And so I think it's quite remarkable that over the course of evolution, we're able to establish these interactions and contribute to the efficiency of catalysis. So using design and experimental evolution, we've been able to take a primitive binding protein, a helical bundle that has no inherent affinity for the substrates, much less activity toward converting them to a specific product, to completely control the reaction pathway and stereochemical outcome of a abiological Diels-Alder reaction, here giving rise to this Hetero Diels-Alder product, a very valuable dihydropyran in greater than 99% enantiospecificity. So it turns out that this scaffold is remarkably pliable, very malleable, and much more so than we would ever have anticipated. So I told you that we've been able to catalyze ester hydrolysis with very high catalytic efficiency. We've been able to do a bimolecular Diels-Alder reaction, and still unpublished work, we can do hydride transfers, so enone reductions using Hunch ester. And we've also been able to do retro-aldol reactions, all of which are dependent on metal-ion catalysis. So the metal-ion serves as a Lewis acid to activate the substrate or water to promote these individual transformations. There's no reason that we should be limited to zinc. In fact, in his original studies, Brian Kuhlmann had shown that MID1 could also bind to other metal ions, manganese, copper, etc. We wondered whether we could go even farther away from zinc and look at gold. Gold catalysis has undergone a renaissance. There are lots of interesting chemical transformations that are promoted by gold. And in a collaboration with Anastasia Alexandrova at UCLA, her student, Michael Neshe, had made a variant of MID1 that we were interested in. And in this variant, there are two methionines that are sitting at the bottom of a hydrophobic pocket. And my students, Sabine Studer and Rafi Frey, wondered whether or not this protein would be able to bind a gold 1 atom. And that could be interesting because this could react with substrates like an ethynyl aniline and do a hydroamination reaction to give indole. In this particular case, you go from a non-fluorescent starting material to a fluorescent product. It's a reaction that Tom Ward at the University of Basel suggested to us. This is supposed to work by substrate binding to the active site, displacing one of the thioethers to give a gold complex. The gold interaction with the triple bond activates it for nucleophilic attack by the aniline nitrogen to give a vinyl gold adduct. So it's a covalent gold carbon bond. And that is resolved by proto-demethylation, likely the slow step in this overall process. So the starting MAL1 is what we call the variant with the two methionines, is able to promote this reaction. But you can see that it is a very slow process. After 48 hours, we convert relatively little of the substrate, which is this peak, to a little bit of the product. However, we believe that that activity is dependent on the protein, because if you mutate these two methionines to leucines, you lose the ability to do the hydroamination. If we replace one of the methionines, this one, at position 87 with a cysteine, then we actually improve activity quite substantially, likely because we're fixing the gold very specifically at the active site. And Sophie Basler has been able to evolve this, in preliminary experiments has found an additional mutation that can further improve its activity. And this is a leucine mutation right at the active site to an alanine. So Jack Fuller in Anastasia's lab has made a model of this system. This is the vinyl gold adduct intermediate that we think is probably the block in the overall transformation. But you can see this fits into the scaffold very, very nicely. The leucine that was mutated to an alanine by Sophie is sitting right above the gold. And so you can understand how it might be able to influence the chemistry. And so we're very excited about the possibility of finding additional changes in the system to further improve its activity. So I'm just about at the end of the lecture. What I think the take-home message is that these helical bundles are remarkably versatile in terms of their catalytic potential. They can carry out diverse chemistries ranging from simple hydrolysis reactions to abiological cycloadditions, aldo reactions, hydroaminations, et cetera. I think we've just begun to scratch the surface of what these proteins will be able to do. What's also striking to me is that there's relatively little chemical information required. As long as you have a little bit of starting activity, you can optimize through repeated rounds of mutagenesis and screening. Maybe this is a reflection of the choice of scaffold, and so it will be interesting to explore other computationally designed systems. In fact, I'm sure that all of you are aware of the advances in computational design, starting from Steve Mayo's pioneering work on zinc finger proteins to David Baker's recent tour de force and generate a palette of completely diverse structures, all prepared with atomic accuracy. So together with David, we've been trying to functionalize some of these with metal ions. For example, we recently were able to make Tim Barrel variants that contain lanthanide atoms. This was just recently published last week in PNAS. Sophie Basler in my lab has also been equipping these helical bundles, these triplexes with zinc ions. And she already has a very efficient Diels-Alder catalyst that gives preferentially the opposite enantiomer of that shown in the mid-1 based scaffold. So I think there's a lot of exciting things to expect in the coming years. Simple strategies for introducing metal ions into proteins can provide us a huge range of novel activities that we can hopefully take in useful, practical directions. So with that, let me thank the people that did this work. So the stars of this show are Sabine Studer and Sophie Basler, who really did all of the evolutionary studies and most of the kinetic characterizations. Juske Otte has further diversified the mid-1 scaffold. Anna helped with the Diels-Alderase at the beginning. Here, Rafi was important for the gold chemistry, and these are the people that helped us crystallize the proteins. We also benefited enormously from great collaborations in this project, initially with Brian Kuhlman at UNC, Ken Hauk at UCLA, and Anastasia Alexandrova also at UCLA. So I'd like to thank all of them for their incredible enthusiasm, hard work, and enormous contributions to this project. I'm very, very grateful. So with that, thank you, Roberto, for the invitation. It's a great privilege to be able to present this work to the audience today. I'd be happy to take any questions.
Thank you, Don. That was beautiful work. So I can already see that there are questions that have been typed for you during your presentation, so you can go ahead and look at them.
I have to end the show?
It's already been ended.
Oh, okay. Okay, questions. For the stability calculus, those are not my questions.
So you can go to the fourth question.
From Jacob Wolf. Enzymes under 100 amino acids in nature are relatively rare. In what ways do you think that small enzymes are advantageous for enzyme design? And what are the disadvantages of smaller enzymes? the advantages in terms of evolution is that you can pretty much explore a much larger fraction of the possibilities than you can with larger enzymes. The disadvantage is clear. I think most larger enzymes benefit from much greater stability. And also the possibility for regulation. That's going to be harder in this case. We've done some preliminary NMR studies of the Diels-Alderase, and it's very clearly a molten globule. Nevertheless, it's a very active molten globule. It's surprisingly active. So I think those are the trade-offs that you have. You have a lot of flexibility, and perhaps maybe that's one of the reasons that the system seems to be quite promiscuous with respect to substrate scope. But there will be disadvantages, namely stability. The second question is, if these relatively simple structures can be improved to comparable efficiencies, why did natural proteins with analogical functions often evolve such complex structures? I think that question is related to the first. Probably you want to evolve for greater stability, but also the possibility of regulating activity. I think those are really the big advantages of modern proteins. It's not necessary when you're doing in vitro studies on studying a single residue, a single reaction, but in a complex environment of a cell, there are many, many other considerations that have to be taken into account. The next question is how a monomer protein can be engineered to dimer in vitro cloning. This type metalloprotein engineering can be applied for longer proteins. Okay, I'm not certain what the question is. But if the question is, can you apply this approach to larger proteins? I think the answer is yes. So the trimeric helical bundles that the Baker lab has made, very, very long extended structures, are much larger than MID1. And we have, for evolutionary purposes, connected the chains, which required some additional engineering. And that has worked very, very well. I mean, what you want to make for homo oligomers, you want to diversify the sequence just to make the evolution a little bit easier, make cloning the variants and whatnot. But I don't see any size limitation there. The next question is, since not all mutations can be tested in directed evolution, is there a rational way to bias mutations to get intended activity? This is from Liz Meyering. Hi, Liz. Yes, this is possible. And in fact, this is what we do usually. What we have found is that the biggest effects usually originate from mutations at the active site. That makes a lot of sense. You're going to get your biggest bang for your buck if you're close to where the action is. And that is both in a positive sense, but also in a negative sense. So mutations at the active site can also be highly deleterious. But so as a consequence, we often will first start by saturating each of the residues that line the binding pocket to find the best choice at those sites. That has been an effective strategy, but you also probably miss a lot because you don't get synergistic interactions. And there, it would be much better to screen, to have a higher throughput assay, which is why I'm very excited about the microfluidics. In the microfluidics system, we can look at libraries that contain 100 million variants in a single experiment and really sample a lot. That means you can randomize 6 residues simultaneously, completely, and look at all combinations. We recently did this in an engineering exercise to change the substrate scope of a natural amine oxidase, a flavin-dependent enzyme. And there we were able to completely remodel the substrate preferences in a single experiment, which I think is really amazing, without losing any catalytic efficiency. So this is a really powerful way to go. And I think we just need comparable methods to go forward. So our droplet sorting requires fluorescence, and so you need a fluorescence readout or to be able to couple your reaction of interest to a transformation that leads to fluorescence. Have you tried to include indel mutations to improve some of your metalloenzymes? Not on purpose, but sometimes they come up. But we have not really explored this as an option. I think this is also an area for future exploration. I think going back to the computational methods, though, will be probably more successful, because in computation, you can look at orders and orders of magnitude more variance than we can ever look at experimentally. And so if computation can explore this indel space and tell us where to focus our mutations, going back to Liz's question, I think that would be incredibly powerful. So this is a question from Nick Polizzi. Great talk, follow-up on stability. How does evolution change the TM of mid-1? I assume the change in crossing angle creates more context between the two helix pairs, so probably it becomes more stable. Is that true? We looked at stability, and it's complicated because we remove one of the zinc binding sites. And so we actually destabilize the protein initially. It may have become a little bit more stable in the optimization, but I don't think it was a big effect. Follow-up question was, do you see this trend toward more stability in general? And all of our other evolutionary experiments with enzymes, we have indeed seen an increase in stability in step with optimization of activity. But those were based on natural protein scaffolds, which already were inherently very stable. When we introduced mutations into the active sites, we often lost activity, but during the optimization, we not only improved catalytic efficiency, but we would improve or bring back the stability. Maybe not all the way back to the original protein, but we recovered quite a substantial fraction. Another question by Nick. Lastly, you just hinted at this, but what do you think the role of protein dynamics plays in catalysis in these proteins? Your X-tau structures are very nice, but this is lock and key picture capturing most of the relevant aspects. Yes, that's true. Dynamics is certainly important. This is also true with the other computational designs. And here I'll give a shout out to Roberto, who has just published a very nice paper in Nature Communications studying the chempeliminase, doing room temperature crystallography on all of the variants that we had published, and showing that the conformational landscape changes as you evolved, and this is really important for activity. It's almost certainly important in these helical bundle proteins. We don't see very much product inhibition, and I think it's because these things are incredibly dynamic. As I said, the metal-ion doesn't bind very tightly, and in fact, we suspect that the metal-ion is brought into the active site during catalysis, that the substrate is absolutely crucial for creating a catalytically competent function. But when the reaction is over, everything can fall apart again and then restart. Whether that's true, we still have to test, but I think it's an intriguing idea. In general, I don't think dynamics is so important in the sense of reinforcing reaction modes, but it's clearly crucial for creating an environment that increases the probability that reaction will occur in a productive fashion.
And Don, if I may, because that's a very interesting question, do you think this scaffold could also be used for catalysis of reactions that do not involve a metal ion?
I would guess yes, but we haven't explored that. We've been focused really on taking advantage of the intrinsic activity of the metal ions. Just because they bring a lot to the table, might be hard. Well, I don't know. I don't know. If you put a reactive lysine into the active site, I'm sure that it will do organocatalysis. But I also the possibility of introducing organocatalytic groups into these pockets would be quite interesting because they do chemistry that's complementary to what one sees in other systems.
Yes, and if you could achieve a higher activity from that scaffold over the natural scaffolds that you evolved previously, that would also be interesting as an application.
Yeah. I don't know how well these will compete ultimately. My guess is more stable scaffolds will be better. That's just my instinct, but maybe I'm wrong. I'm surprised that a molten globule can be so active. I mean, we had shown this some years ago in a circularly permuted, or in a conformationally redesigned chorus-mate mutase, but seeing it in these artificial systems gives me great confidence that this is an interesting way of going forward. Next question, can zinc be replaced with other transition metals? The answer is yes. Are these designs have any promiscuity to other metal ions outside the bacterial cells? We typically isolate the proteins from the cell, purify them, and then add we extract any metal ions that might be present using EDTA, and then add back the metal that we want. So as I said, Brian had already shown that mid-1 will bind copper, manganese, and a whole range of different ions. And this is also true for mid-1. Most of these don't have any interesting activities, but we haven't really explored them very extensively either. I think this is redox chemistry would be particularly interesting in this regard. Manganese would be a great system to pursue further. And then the last question, alongside substrate affinity, how did the zinc ion affinity change throughout the evolution? That I can't answer because we haven't done detailed measurements for the various variants along the evolutionary trajectory. We know that the final variants typically don't bind it very tightly, often in the micromolar rather than the nanomolar range. But in the presence of ligand, for example, in the esterase, when the transition state analog is present, the affinity is in the nanomolar range. So clearly, it's context dependent. Didn't Alan Firsch propose that natural, whoops. There was a question, didn't Alan Firsch propose that enzymes evolve through molten globules? I don't remember Alan saying that, but it's possible.
Okay, great. Thank you, Don. Please join me in thanking both of our speakers for wonderful presentations. And before you leave, I will mention again the next round of webinars. So we've had an excellent first round of 4 webinars throughout the fall. I hope you enjoyed them. We have an exciting lineup of future speakers. So if you allow me, I'll share my screen to show you again that slide and remind you that you can find that information on the PEDS website. Okay. Here we go. So if you can see this, we will be restarting in January. More details to be provided for that particular webinar. But if you want to stay up to date, please check out the PEDS website here on the top right and the PEDS Twitter account at the bottom right for all information related to the webinars.
Thanks, Roberto.
With this being said, I just want to thank everybody again and see you next time.